Okay, yeah, recording has started. Okay, so till now we are doing protein and ligand based docking. Okay, so like you have already practiced this thing, how to do docking using Autodock Vina, and I would like to know how many of you created accounts in Haddock. Yeah, please raise your hands now if you have created account. Only
Yes,
four.
sir, we created. We are waiting for it to activate. So,
No, it will get activated immediately. Okay, you will receive a mail. You
yeah.
have to click on the mail. It will be get activated immediately. Okay.
Yesterday, when I was using it, it says that wait for us to get back to you.
Okay.
Yes, sir. It takes almost 24 hours to get activated.
Okay.
Yesterday, same thing happened in my systems.
Okay. They might have changed their set of rules because they are in process of upgrading many tools actually. Okay. Mm, then, okay, no issues. So today, so till now, like based on the previous class, you might have understood like how we use the secondary metabolites of the plants or the drug molecules synthesized by the chemist, uh, how we use them as a drug molecules to tackle different biological problems or different diseases, disorders. Okay, so that's what we have discussed yesterday in the first hour. Okay, so today let's talk about the peptides. Okay, what is a peptide? Yes, what is a peptide? Yes, have you heard this word before, peptide?
So it's a chain of amino acids, sir.
Yeah, it's a chain of amino acids. So again, the protein is also a chain of amino acids. Then what is the difference between peptide and a protein?
So the length of the chains, peptides are shorter chains, whereas proteins are longer chains, and they also have various structures.
Yes, yes. So peptides are the small ones, like usually if amino acid chain of even 10 amino acid chain, that can be called as a peptide. So like we have around maximum of 50 to 80 amino acids. That is the maximum size. So even if we have around 15 to 30 amino acids, that can be considered as a peptide. Even 10 amino acids also we can call as a peptide. Okay. So these peptides, we can also use the peptides to target the proteins. Okay. So so today we are going to have one practical session. Like the problem, it's you can consider it as a case study. So as we were discussing, so like we will start with a problem. Okay, so we can use these peptides to target the proteins of our interest. Okay, for some particular problem. Okay, so let me connect the point. So we can both target the human proteins using the peptides, as well as we can target the microorganisms like bacteria also. Okay, so to inhibit their growth or to stop the film formation. Okay, so that the bacteria won't multiply or they cannot infect the host cells. Okay, Yeah, suppose let's assume we have a protein. Okay, so this is some protein. Let's assume this is the outer surface of some particular protein, and this binds to the, let's assume this binds to another protein. Okay, so this protein, so it will bind to the another protein. Okay, so this is the another protein, it has to bind. So now we don't want this binding should to happen. Okay, we don't want this binding process to happen. So how we are going to tackle this? Okay, we can see here this region. So this is the region which is going to bind with the other protein. So can you use a ligand here, because there is no pocket? Okay, it is like completely outer side, so we cannot use a ligand molecule, or we can give a try also. There is nothing wrong in giving a try. And but here in these cases, so as this is going to target a protein instead of, so you should, till now what we have discussed, like we have an enzyme and it binds to a substrate. Okay, then it leads to the formation of product. So this is what we have discussed. So instead of the substrate, we are replacing this one with the ligand. So that is what the strategy we have followed for the enzymes. But this protein is not an enzyme. Okay, this protein, let's assume this protein is not an enzyme. So it is just going to interact with another protein, and when it interacts with the other protein will get activated, and that will carry out some signaling process inside our body. So we don't want that association to happen. So this protein we don't want to interact with the other protein. Okay, so now here we can design a peptide. Okay, let's assume all these circles are the amino acids. We can design a peptide as such. It can bind to it and it can block the, so once this peptide is bound to this region, so it is not available to bind to this the protein 2. Okay, so this first protein, this cannot bind to the second protein because of the intervention of this small peptide. Okay, so suppose it binds to the phosphate region, let's assume. So this will bind to the protein, and this second protein has a phosphate at this location. Okay, and it will bind specifically to this phosphate. So now in this case we can use a phosphopeptide. We can use a peptide which is modified as a phosphopeptide, and that can go and bind to this location. Okay, so that is what we can do actually. Okay, so now let me show you the same thing. I hope this concept is clear. Like whatever I have explained now, is it clear for you all? Okay, so like, and more for most of the teachers, okay, so breast cancer is one of the favorite word, what I feel. Okay, so if we talk about the breast cancer, do you remember, have you heard this word from your teachers before, the word breast cancer? Yes, so whenever they say breast cancer, what are the gene names they will that will come out of their mouth usually? Yes, ma'am, do you have any idea, like the genes associated with the breast cancer? No. What about others? If I tell the names
So is
now,
it
then
one?
yeah, yes,
Right,
Soumya, it
yeah.
is BRCA1, BRCA2. Yes. So like BRCA1 and BRCA2 are the popular genes. Like it is usually in the on the tongue of the most of the teachers and most of the people who talk about breast cancer. Okay. So now let's see an example of the breast cancer. Okay. So let's first see. I want to work on the breast cancer. So I want to look at the sequence of the breast cancer. Okay. So I'm just going to UniProt and typing BRCA1. Okay. So we want BRCA1 of Homo sapiens. You can see. So this is the third one is the Homo sapiens T38398. Just click on this link. So you will be directed to the page of the BRCA1. So now here we want the sequence. Just click on the sequence. Okay, if you want to understand, Seema, first and most important thing is that if you want to work on any protein, first you have to read the research paper or review article associated with that paper. Okay, so now here we don't have that time to read the papers. So I'm going to tell you some methods. Okay, to it is not like if you do this you cannot read the papers. This is not the end, just for a quick thing. Okay, whatever we are going to do now, so this is like a quick thing to understand about the protein, but you have to read the research paper. Okay, if you are working on any project, or in future if you want to be in research, whatever the disease or target protein you are working, you have to know in and out of the protein. Okay. Like if it is like insulin, a small protein, we should able to tell the entire sequence of the protein. Okay, that much memory or love you need to have towards your uh, project. Okay, or the problem you are trying to address. Okay, so this is the entire protein sequence. You have, I think you remember it's around 1800 amino acids are there. Okay, I've just copied this one. So let me save this also. See, whenever you are doing any project, data is very important. Okay, so this whatever the information you are accessing, you have to save that information. So I'm just pasting this one. So I will save this one. So downloads. So I'm typing it as BRCA1. Okay, so this is the sequence, right? Protein sequence. Okay, if you want to save in FASTA format, you can type .fasta, so it will be saved in the FASTA format. Okay, because here we have the header line. Okay, this means it is a FASTA format. So now we have the sequence. So as per as the theory what we have discussed now, so this protein BRCA1 is also similar to this. It will bind to some protein which has been phosphorylated. Okay, so to confirm where this phosphate binding region is present, so we have to copy the sequence, then go to interpro. So why we are using interpro? Yes ma, what is the use of interpro?
To identify the domains.
Yes ma, yes Nikhil. So we are going to identify the domains. So paste the entire sequence here. Okay, I have pasted the entire sequence of BRCA1 here, and now just click on the search. Okay, so I am asking you guys also, let the search go on. Okay, the search is in process. Meantime, so now before this I was telling you guys to read the research papers. Then you may ask, sir, how to find research papers? So you are very new to this, all these things, research particularly. Then how to find the research papers? You can see here, if you come to the top, okay, So as this BRCA1 is the most favorite gene of many people who are working on breast cancer, you will have many publications. Okay, so you can see E3 ubiquitin protein ligase that specifically mediates formation of lysine 6 linked polyubiquitin chains and plays a central role in DNA repair by facilitating cellular response to DNA damage. Okay, so these are some papers associated with this thing. Okay, so you can see this BRCA1 and BRD1 heterodimer coordinates a diverse range of cellular pathways such as DNA damage repair, ubiquitination, transcriptional regulation to maintain genomic stability. So like this we have, you can read all these things, and you can see BRCA1 is making associating many proteins. So BRCA1 and BARD1, and here I can see something else, BRCA, this is related to BRCA2, and you can see BRCA1 and RBBP8. Okay, so you can read, and all this PubMed, these are the research articles associated with this thing actually. Okay, so this paper is saying BRCA1 acts as a transcriptional activator. So these are 15 publications. Okay, and you can also see here there is an article saying BRCA1 in this serine 308 plays a role in normal cell cycle G2M transition. Okay, so but this has been withdrawn, this they have eliminated this one. Okay, so in 2026 I don't know what resolution you have made, but this Uniprot people, they have made a very huge decision. They have made a lot of changes in their website. Okay, on the data available on their website. Okay, so you can see all the publications here linked to this BRCA1 and with their specific functions. So if you don't find anything related to yours, then you have to give a search, and you can see here based on the domain. So DNA binding, this is a source, but there is no publication. But for DNA binding, we have one publication. Enzyme binding, we have one publication. So this ubiquitin ligase activity, we have a publication. Identical protein binding, and it binds to P53. For that, we have one publication. And RNA polymerase binding, we have a publication. So like that, you can see many papers publications listed here. Okay, so okay, so you can go through the things for your protein and find out what is actually of your interest, and you can select the paper and read. So this is the easiest way to find the publications, okay, linked to your protein of interest. And now let's move on to the results. Okay, the results are ready. So you can see here. Okay, so you can see this is entire family, it is saying BRCA1. This is the zinc or ring finger domain. It means zinc or ring finger domain, it is usually associated with the DNA binding. Okay, simply you can type zinc or ring finger domain in Google plus DNA binding, you will see a lot of papers. Okay, and we have this one, serine rich region associated with BRCT. So this region, it will make association with the another protein called BRCT. And we have another region, so this is also BRCT domain. Okay, so this is also BRCT domain. So these are the regions associated with the another protein called BRCT. Let's see here whether they have mentioned BRCT anywhere. Okay, you can see here interacts with ABRAXA1 phosphorylated form via the BRCT domain. Okay, ma. So you can see this region is called as BRCT domain. So this BRCT domain interacts with this protein called ABRAXA1, which is in the phosphorylated form. It means here as we have discussed the theoretical aspect here, so we can use a phosphopeptide to block the region of BRCT domain so that it will not bind with this ABRA XAS1 protein. Okay, so this is important for recruitment to sites of DNA damage. Okay, so we are stopping some process here. Okay, so why we are stopping the process, really, to be frank, I don't have any idea. Just to make sure, make you clear, the understand the concept, I am taking this. Okay, and now you don't ask me why I am targeting this one, because I don't have any agenda with respect to that. My own agenda is that to make you understand why we are using phosphopeptide here to block this region. Okay, so this BRCT domain region of BRCA1 binds with the phosphorylated form of this one. So that is the reason I am using a phosphopeptide to block this region. Okay, and if you are working on it, then it is your responsibility to know if I block this, okay, association, what is going to happen, how it will affect the cell. Okay, that will lead to apoptosis or not. Okay, suppose you are targeting cancer, so if you block this one, so will it lead to apoptosis? Okay, if it is leading to apoptosis, blocking this association is the best choice. Okay, so like that you need to read the theory. Is it clear for you all what I am trying to convey? Okay, Varun, Akshaya, Zohab, Naveen, Pravalika, Soumya, Caroline. Okay, ma. Thank you guys, you are listening. Yeah, now we are going to target this BRCT domain. So now what you will do, make a note of this BRCT domain regions. Okay, so let me take another notepad file. So now I understood that, so this BRCT domain is the important region, okay, to target. So what are the sites it has? So the first one, so this is from the PFAM, it is 345 to 508. So 345 to 508. Then we have this region 1608 to 1753, and this is continuation till 1859. Okay, well, let's take a continuous thing here because both of them are side by side. So let's consider it as a continuous region. Why it has not struck? So this is from 1608 to 1. I just want this second one. Okay, 1859. So 1859. So these are the BRCT domain regions. So let me save this info also. Another thing is that you can take a screenshot of this output, okay, and you can showcase in your result part. Okay, you can download the picture or you can take a screenshot is the best option here. Okay, but as you cannot see the exact numberings, that you need to make a note. Okay, now I want the, so this is the primary analysis part we are done. Okay, now we want the tertiary structure of the protein, 3D structure of the protein. So where you will find that one? So for the screening, we are going to use UniProt. From there, we will go to the protein data bank, right? So come to the structure. Okay, you don't want the entire protein. Okay, ma. You just, even if you have the small fragments, so those fragments are enough for docking. Okay, so what is the 305 for 345 to 508, the entire size? Let's calculate. So 508 minus 344, this peptide is 164, this domain is 164 amino acids long, and this second one is 1859 to 1607, 252 amino acids long. Okay, so I think this is the good size. Okay, so now let's look whether we have this region structures are available or not. Okay, so you can see here we have a peptide for 1 to 110. Okay, this is not our interest region of interest, and you can see the second one we have from 1646 to 1859. So is it falling? This is falling into the this region. Okay, so you can see many things here. So many proteins. Okay, 1646 to 1859 we have many structures, because based on our analysis using interpro we can say that, so this is the important region. So that is the reason people across the world also are working on that region. Okay, so it's not like discovery of an penicillium antibiotic. So we are not in that stage now. Okay, random discovery stage. So that was done in 80s and 1990s. Okay, before 1990s. Nowadays random works uh, are not at all going to do anything. Okay, we need to be very specific at a problem of our interest, and we need to know exactly what we are doing, why we are doing, and a lot of data is available online. You have to analyze the data, and then you have to draw your primer. You have to analyze the data, and you need to make your proposal or idea more strong based on the available data. Okay, now based on the available data only we have concluded that that these two regions are important. Okay, by doing a small analysis, right? So let's check whether we have any peptides. So you can see this is alpha fill, it means that is a model. So all these are when modeling is start, all are the modelings one, those are not required. So we have to check for the regions which are determined experimentally and which are falling in the region of this one 16462 to 1859. And what was the resolution, should be less or more? Yes ma'am.
Let's
Yeah, resolution should be less. So now let us look at which one is having less resolution. So I can see this one, it is having 2.5. So we'll see whether we can find less than that. So we have one which is 1.75. So let's see whether we can find less than that. I hope everyone is following and I'm not rushing. Okay, 1.75 was the least one. So that is the least one. Okay, and we have one another one is 1.85 also. So first let us go and check the 1.751 in the PDB, whether the structure is good or not. Okay, this one. So click on this PDB, RCSB PDB. So that will take to the protein data bank. And you need to make sure this region does not have any what we can say mutations. Okay now, so you can see mutations are no. So this protein they have expressed in the E. coli, but it belongs to Homo sapiens. And you can see the structure, there are many Ramchandran outliers. Okay, so that structure is not that good. Okay, so what we'll do, we'll check the 1.8 by 51 also. So I hope make a note of the PDB IDs. Okay, when you are working, everything you have to write in your notes. Okay, so what is the PDB ID of 1.751? How many of you remember the PDB ID? Yes, no one. The PDB ID is 4IGK. Okay, 4IGK. So it's like if you are working on some protein, if you have selected that one for your studies, even if a person asks you by waking up all of a sudden in the morning, you should be able to tell the PDB ID. Okay, that much dedication and love you need to have towards your work. So this one we have 1.85 wala 1T15. Okay, so if anyone is having doubt for that 1.751, you can look here. So this is 4IGK. Okay, we'll go to PDB plus 1T15. And you can see this has some mutations now. Okay, but and other thing you can see this structure is also not that great, so it means and it has a lot of clash scores. It means clash, what is mean by clash score? If two amino acid side chains are overlapping, that is called clash score. Okay, so comparatively that previous one itself is more better. Okay, you can compare this worldwide PDB validation report of the two or more PDB IDs of a protein, and then you can decide which one you have to select. Okay, so I'll take another tab, PDB plus 4IGK. Okay, you can see the class score here. It is on the, it is in the center, but for this it is towards the red side. It means this 4IGK is only the best one. Okay, so now the another thing, so we are done with the protein selection part. Okay, and now we need to check, see this sequence whatever we have, you should see here the PDB is available from 1646 to 1859. So let me make a note that one also here, 1646 to 1859. Okay, so what is the total size? 1859 minus 1646, it is 213. So 213 is not a small number. Okay, 213 is a big number, and within that 213 also we need to know exactly where the second protein is going to bind. Okay, so how we will do that? We need to check the protein, it is bound to any other phosphopeptide. Okay, now so luckily you can see here the structure whatever we have selected. You can read the title, structure of human BRCA1 BRCT domain. It is in complex with ATRIP peptide. Okay, so it means where the peptide is binding, that is the actual region which is involved in binding with the other protein. Okay, so because we have to believe in this thing, because this is the experimentally derived one. Okay, that is the reason we have to believe this. And you can read this paper in detail. Whenever you are selecting any PDB, you have to download the research paper with associated with it and read it in detail now. Okay, so if you scroll down, you can see we have this BRCA1 and two chains of 214 amino acid length and no mutations. And if you scroll down more, you can see this is the peptide ATRIP. So the peptide length is 7 amino acids only, and this also has no mutations. Okay, so what I was saying, we have to target using the phosphopeptide. So now we have to check whether this ATRIP is a phosphopeptide or not. That we have to cross check. Okay, so based on my understanding, I am telling we have to use a phosphopeptide to target. So these scientists, the researchers who have worked on this, they might be also working on a phosphopeptide only. It means ATRIP peptide will be a phosphopeptide. It is not a simple peptide. What is a phosphopeptide? For the amino acid, you are going to substitute one functional group with the phosphate group. So then it is called phosphopeptide. Okay. So let's open this structure in Pymol and see where exactly it is binding and whether it is a phosphopeptide or not. Okay. So open Pymol. You can also do along with me simultaneously, or you can listen as of now. Okay, in the next hour, anyway, docking part, once your accounts are activated, you have to do this docking. Okay. So what is the protein? What is the command we have to type? Yes, anyone of you remember? So I want to open a protein here in Pymol. What is the command?
H.
Yes, Nikhil answered. It's fetch. FETCH in small letters, and you can type either in small letters or capital letters. The protein ID 4IgK, either in capital or small. Anything is fine. So fetch 4IgK. You can see the protein is visible here. Okay, and you can also see one more thing. It is having two protein chains. Okay, so click on the sequence. So you can see the sequence here. It is from starting from 16462. Scroll towards the right hand side, and some 3 amino acids are missing here, and it is ending with 1859. Okay, and then we have the B chain. It is also the same, and you can see this is the peptide ACSCPPQFG. So this is the peptide, and this is the phosphate. Okay, serine, the amino acid serine has been phosphorylated. Okay, what is the 3-letter code for serine? SER. So as the serine is phosphorylated, it is written as SCP here. Okay, so you can see the scientists who have worked on this, they also used a phosphopeptide, and this GOL, it is the solvent. Okay, and zeros are the water molecules. So now, as you all know, we have to do the protein cleaning part. So what is the first step in protein cleaning?
Remove the water molecules.
Yes, remove the water molecules. So click on the A, actions, and click on remove waters. The water molecules will be deleted. And what is the next step? Yes, you people have done this thing too many times, at least 1 or 2 times. Okay, tell me what is the next step now? So apart from the protein, what all we have here?
Sir, check
Yes,
for any missing amino acids.
that is the last step. So we have, see, Nikhil, Ramesh,
So checking
Bhavin,
for atoms, zinc atoms like that.
uh, what we call them as heteroatoms. Okay, you can see
Yes.
this GOL, this is a solvent. This is actually not required. So you can select this GOL and we can delete. Okay, actions, remove, remove atoms. Okay, so they are deleted, and you can see here we can delete the peptide also, but I want the peptide to be here because we have to define the grid parameters, right? So for that we are going to use uh, peptide, and we have another thing is additional protein chain. Okay, we have two peptides here, two domains, two chains here, A chain and B chain. So one chain we can delete. Okay, so I'm going to select chain B and delete it. Say SELE space chain space capital B. So this will select the B chain and I'm deleting it. Actions, remove atoms, and you can see this is the peptide associated with the B chain. Okay, so we have to delete this one also. So I think this is chain D. So select SELE chain D. You can see that has been selected and I'm deleting that one also. Okay, now we have only our protein and our peptide molecule here. Okay, one protein and one peptide is here. And as Zoeb said, we need to look at the missing residues. And you can see here, there are some missing residues here, TED. Okay, and you can see there is like line, discontinued line here, right? Can you, can you see the discontinued line? Yes, so that discontinued line is the one specifying the missing residues here in the structure. So we can add these missing residues, okay, because or you can ignore it also. You can see this is slightly away from the binding site. Okay, so you can rotate and see, is it in the binding site? It is away from the binding site. Okay, so it means we can ignore that region. Okay, or you can add that region also. So if you want to add, you can go to the legacy plugins. Okay, this plugin button is not working. Okay, you can select this missing residue. This is T. T means, what is T? T means Trivanima. Okay, okay, let's look at that process later actually. Okay, this plugins is not working. Okay, that's a lengthy process actually. So as this is away from this site, okay, we can ignore that one. Okay, so now what we are doing, we are going to save this one. Okay, so file, export structure, export molecule, then click on the PDB options. Okay, then click on save. So go to your folder where we are saving this. So we are saving it in downloads, BRCA1. So here you type it as BRCA1 hyphen P. Okay, it means peptide. And the save as type, we are saving it as the, we are going to save this one as the PDB format. Okay, so select the format as save as type PDB. So it will be selected in the PDB format. It will be saved in the PDB format. Okay, save it. So now this one is for evaluating the grid parameters. So we want the other one also without the peptide for doing the docking. Okay, so for generating input files for the docking. So for that we have to delete the peptide. You can see the peptide is with the C chain. You can see in the sequence peptide is shown as the chain C. So let me select the peptide SELE chain C. So the peptide will be selected Okay, you can see the peptide has been selected here. And actions, remove atoms. So the peptide has been deleted. So go to file, export structure, export molecule, then PDB options, then click on save. Okay, so this is the BRCA one without peptide, so I'm simply saying it as BRCA one and PDB. Just save this one. Okay, so now both the files has been saved. Okay, so what do you want? You have protein now. You want a ligand also, right, to do the docking. So the existing peptide, we have already deleted that one. Either we can use the existing peptide as of now for our docking purpose, okay, or we have to download the phosphopeptides. So just go to Google and type phosphopeptides plus database. Okay, so you can see we have a peptide atlas. Mm, so these are the different websites where we can find the phosphopeptides. Just a second. And whatever the information given here, it is may be correct or may not be correct. You have to read everything and you have to cross check. Okay, now so you can see this EPSD is a different one. It is a phosphorylated site database. None of these websites are related to phosphopeptides. So you can see here we have this MPPD database. It is containing unique phosphopeptide. 3457 unique phosphopeptides are available in this website. So click on browse. Can you find any option more where we can see the list of peptides? Okay, but this is something like they are not displaying any peptides here. Okay, visually we don't have any database for these phosphopeptides. We have to create them. Okay, so uh, like we can create some of the phosphopeptides. So as of now, we can use the peptides, the the same peptide here. Okay, but we have the peptides for antimicrobial peptides. So we call them as AMPs. You can see antimicrobial peptides, antibiofilm peptides, TB peptides, protease inhibitors. So this is one of the database which we can rely. Okay, you can see different peptides, anti-inflammatory peptides. This you can use. I think we can go for some protease inhibitors, and you can see these are the different proteins. AP. So this is the uniprot ID. So we have 55 inhibitors, and this is the sequence, but this is not a phosphopeptide. So like that, you have to check some database where you can find the phosphopeptides, and that you can use for our current work. Okay, so as of now, so this is one website you can go through. I'll just drop a link. So this is one of the domain which is currently which is the work, huge work is yet to be done. Okay, so that is the reason we don't have any proper databases too. Okay, so coming back to this thing, let's take the same peptide from here itself, and we will dock it back to the same thing. Okay, so for that, what we are going to do, so I will show you a different method now. So I'm not going to do any cleaning, but I will save only the peptide one. Okay, so focus here. So I'm opening the same protein, fetch 4-IgK, and we know the chain C is the one with respect to the peptide. So I'm going to select chain C, select chain C. So you can see the chain C has been selected. Okay, so you can see here only the chain C is selected. So in this selection, rename it actions, rename selection. So this is the, I'm typing it as PP, phosphopeptide. Okay, now I want to save only this PP. So to do this, the same process we'll do, export structure, export molecule, and you can see here there is option called selection. Here you just select PP, and then click on all the options here in the PDB options, then click on save, and only the peptide molecule is going to be saved. PP, I will type the small pp, and save as type PDB, then click on save. Okay, so now I have saved the molecule. Let's do a cross check whether we have saved only that one or not. So we have the PP. Let's open it in Discover Studio and do a cross check. Okay, you can see here we have saved the chain C, but there are some water molecules associated with the chain C. So that is the reason there are some water molecules also has been saved. So we have to delete this water. Okay, and let's save it again. Click on save, so that will be saved. Okay, so PP and save as type PDB, protein data bank files, then click on save. Okay, and click on replace. Okay, now the file has been replaced. Only the peptide molecule has been saved now. So you can see this one. Can you see this one? This T-shaped structure. So this is the phosphate. Okay, added to the serine. So we'll break here for 10 minutes. After that, we'll do the docking thing. Okay, I will show you how to submit the docking. So as we did yesterday, we are going to use Haddock for this, and most of the times Autodoc Vina doesn't work because we are Autodoc Vina is specially designed only for protein ligand docking. Okay, so here ligand is also a peptide. It may work or may not work. Okay, so that is the reason it's better to go ahead with the Haddock. Okay, so let's have a break for 10 minutes and then we'll continue with the docking process. Okay, so as you are going to use Haddock, we need to know the residues associated with the peptide binding. Okay, within the BRCT domain, which amino acids are playing major role in binding with the peptide. Those amino acids we need to know. Okay, so that we will do now analysis after the break, and we'll set up the docking using Haddock. Okay, so we'll join back exactly at 8:45 and we'll let's continue the session. Okay.